All right, let's get started. So I just wanted to it'll be a few slides on each chapter, and we'll see like themes that run through all of the chapters, and so you can kind of see the connections a little better and have maybe some more context as you go to study for the exam on Thursday. So to start out with chapter 2, we introduced the idea of atoms and what atoms are made out of. So atoms have three main parts. We call them like subatomic particles. So atoms are made up of electrons, which have a negative charge and like very little mass, almost like zero mass. Protons, which have a positive charge. So electrons and protons have like the same magnitude charge, just like opposite direction. So if an electron has -1 charge, a proton has +1 charge. So if you have an atom with 1 electron and 1 proton, it has like 0 charge. So there's electrons and protons, and there's neutrons. Neutrons have about the same mass as protons, and neutrons and protons sit in the center of the atom. So you have those two particles in the center, and then you have the electrons going around the outside. And so we know that atoms are most stable when the number of electrons they have fulfills their octet rule. So different elements will have different numbers of electrons depending on their atomic number, which we'll go over in a little bit. So if they have, let's say they have 6 electrons, in their innermost shell, they're going to have 2 electrons, and then the outermost shell, they're going to want to have 8. But if they have only 6 electrons total, so then they have 2 in the inner shell, and then that would only leave them 4 in the outer shell. But according to the octet rule, they're going to want 4 more electrons. So the octet rule says that they're going to want 8 in their outermost shell, and their outermost shell is always going to be referred to as their valence shell. So I asked you how many valence electrons there are for an element with 6 total electrons. You would say there are 4 valence electrons. But they want to have either 8 valence electrons, or if it's just their innermost shell, then just 2 valence electrons. Does everyone remember those concepts of like valence electrons and the octet rule? With excitement. So atoms will interact with each other to try to fulfill their octet rules. So if an atom, like this would be carbon, if carbon has 4 valence electrons, it's going to try to interact with other atoms to fulfill its octet and get 8 valence electrons. One way that atoms can interact with each other is through covalent bonds. So covalent bonds are bonds where electrons are shared between 2 atoms. So they can be shared equally, and if they're shared equally, that bond is considered a nonpolar covalent bond. And there's really no negative or positive charge on that molecule, the molecule being the combination of multiple atoms. The charge is just equally spread throughout the molecule. In contrast to nonpolar covalent bonds, we have polar covalent bonds. So in polar covalent bonds, one of the atoms is pulling a little more strongly on the electron that they're sharing than the other, and so that the strength of the pulling is the electronegativity. Electronegativity is the strength by which atoms pull on an electron or attract an electron. So if one of the atoms in this molecule is much more electronegative than another, it's going to pull on the electron and have more of it and have like a negatively charged region, which is what we have here when we had hydrogen and chlorine bonding. We have a negatively charged here because the chlorine is much more electronegative than the hydrogen is. So here we have a polar covalent bond. Which side is more electronegative? So the chlorine would be more electronegative because it has more of like the negative charge, more of the electron. Good question. And again, it's the difference in electronegativity that matters. So 2 hydrogens bonding together would be nonpolar, and 2 chlorines bonded together would also be nonpolar. But when you have a hydrogen and a chlorine bonded together, then that's going to be polar. So it's not just like the total electronegativity, it's the difference in electronegativity, because hydrogen has a much lower electronegativity than chlorine, which you don't have to memorize. I will never ask you to like regurgitate which atoms are more electronegative, but that's how you determine whether something's polar or nonpolar. So in addition to covalent bonds, you also have ionic bonds, which are a little bit less common in biology, and that's where you're going to have an exchange of electrons. So one atom is going to be like accepting an electron, and one's going to be donating it. So in this case, you have an electron that went from this sodium to to this chlorine, and now we have a positively charged ion next to a negatively charged one, and that attraction of charges is what's going to be forming our bond. So electrons are on atoms. Atoms want to fulfill their octet rule, so they have to interact with other atoms to form bonds, which help them fulfill their octet rule. And these are two of the main types of bonds that we talked about. And within covalent bonds, we have polar and nonpolar bonds. Any questions about that? It was a while ago. Yeah.
You said you won't ask questions necessarily on like what the electronegativity of this, but you could ask us, is a carbon and nitrogen this bond or that bond? Is that what you can ask us?
No, I mean, I would never. So I would ask you. How would you know if a bond will be polar or nonpolar, and then use the term electronegativity in your answer. And then you would say, I know it's going to be polar if there's a big difference in electronegativity. Good question. Yeah, but I wouldn't ask you to say like which atom is more electronegative than the other. But I think generally you should know that hydrogen has a very low electronegativity and oxygen is very high, and that's why water is so polar. But I wouldn't ask you to compare different atoms. Oh, wrong keyboard. So the last part about chapter 2 was talking about. Oh, sorry.
This is a random question. How long do you expect the lab exam taking?
Like an hour-ish,
Okay.
maybe a little bit less.
And when is that?
That'll be during your lab session, so either Tuesday or Thursday. You look really happy about that answer. Did you think it was another day?
No, I'm just thought like, I don't know.
We'll do a short review beforehand. Okay. So the last part about chapter 2, we were talking about the concentration of protons in a solution. So the concentration of protons in a solution is measured by this idea of pH. So pH, this is a logarithmic scale, and so what that means is a pH of 6 has 10 times higher concentration of protons than a pH of 7. So even though it's only one pH point, it's a 10 times change in the amount of protons. So the lower the pH is, the higher your proton concentration and the more acidic your substance is. The lower the pH, the more basic it is, and the lower the concentration of protons.
The higher the number, the more basic.
The higher the pH, the more basic. Yeah, which means it's a lower concentration of protons. Did I mix it up? Okay. It's like hard. It's a tongue twister for me to say it all at once. Buffers are compounds in our body, or solutions in our body that resist these changes in pH. And so we talk a little bit in future chapters and during the labs about why pH changes are important to, and why we have buffers.
Did we ever go over coenzymes, or is that?
No, we went over that a little bit with chapter 6, and we can review a little bit when we get there. All right, that was chapter 2 in a nutshell. Was that? Should I just have done this to start the class, or this have been
Question.
so much quicker today?
For electronegativity, is that like the one that's more negatively charged?
Yeah. So when you have a molecule, the more electronegative atom will be more negatively charged. But
That one has more electrons, basically.
yeah, once they form the bond, but before they form the bond, they're both going to be neutral probably.
Okay.
Yeah.
So once they form the bond, the one that has control over more electrons is the one that's going to have a higher electronegativity.
Yeah, when you think about it this way, they're both neutral before they form bonds, but one really wants an electron and one kind of wants to get rid of an electron. So the one that really wants the electron is like very electronegative. The one that sort of wants to get rid of it has a very low electronegativity.
Okay.
So then they bond, and the one that wants to get rid of it gets rid of it, and the one that really wants it takes it, and now it's more negatively charged because it took that electron.
Okay.
Yeah.
Got it.
Yeah, great. But they're still like neutrally charged overall. Yeah. Okay, so we have atoms. Atoms bond together. They bond together to make molecules, and cells make a very specific type of molecule called biomolecules. So biomolecules are usually just very large molecules that are synthesized by cells to perform really specific roles. So one of the most common kind of forms of these biomolecules is to have like carbon and hydrogen bonded together like this. That's why sometimes you'll hear people refer to life on Earth as like carbon-based, because a lot of matter has carbon in the center as one of the central atoms. It's very important. And so you often have these carbon and hydrogen bonds. But the thing about carbon and hydrogen bonding is, even though it's very easy for cells to perform and cells to get a hold of materials like that, when carbon and hydrogen are bonded, it forms a nonpolar compound. And these nonpolar compounds are not going to dissolve very well in water. And so we know it's nonpolar because we know that carbon and hydrogen have very similar electronegativities. And since they have similar electronegativities, they just share those electrons equally, and they make a nonpolar molecule. These nonpolar molecules don't dissolve very well in water. So we have to make some modifications in them so we can use these molecules in like water-based systems, which all cells are. All of our body is made up of like 75% water, some astronomically high number like that. So we want everything to be soluble in water to some degree. So we're going to modify these hydrogen and carbon molecules that are really easy for us to make and to get a hold of, and we're going to add these polar groups. So in this case, the polar groups here are these OH groups that cells have added on to make glucose, and now that makes glucose water soluble, whereas if you didn't have these OH groups, it wouldn't be water soluble. So that's the idea of polar groups that are changes to like organic matter like this to make them more suitable to be biomolecules.
I don't know if we're going to talk about this a bit later, but is a polymer the same thing as a biomolecule?
No. So in some cases, yeah. So the term polymer just means like a long repeating unit of like individual, like repeating building blocks. And so you could call proteins polymers. A lot of carbohydrates are technically polymers. DNA is like a polymer. But there's a lot of polymers that aren't biomolecules as well.
So what would be the question
So nothing on this slide would be a question that I would ask, but this was just background to get into some of the more details. Why don't I do chapter 3 one more time, like go through the slides, and then we can go over what potential questions would be. There's 4 classes of biomolecules. The first 3 that we'll talk about briefly, and some of their uses. We have carbohydrates, nucleic acids, and lipids. So carbohydrates were used, we talked about it most for energy storage in the form of glucose, but they can also provide like structural support in the form of like cellulose, which makes up a lot of the structural components of plants. Nucleic acids, another class of biomolecules, they're most frequently talked about with DNA, which is stores like, it's like the instruction manual for your cells. It stores a lot of information for cellular activity, but they can also be used for energy storage in the form of ATP. So I'll talk a little more about ATP. I realized I maybe didn't give the best introduction of what this molecule is in a few slides, but it's actually a type of nucleic acid that helps you frame its function a little bit better. The third class that we talked about was lipids. So lipids are a key component in fat. Fat is used as both energy storage and as insulation. And lipids are also used in cellular structure in the form of phospholipids. So we talked a lot about phospholipids because they have really interesting behavior in water. The biomolecule we probably talked the most about is proteins. So this is the 4th biomolecule. So proteins, in the context of this class, are going to be, we talked about them mainly as their role in catalyzing reactions. So when we have reactions that need to occur at a high rate, to increase that reaction rate, we can use these proteins. And we know from chapter 6 that the protein function depends a lot on its shape. So the cell's purpose for these proteins is dictated by the shape that it can create them in. And it controls the shape of these proteins by controlling its primary structure. So the primary structure of these proteins is the sequence of amino acids that the cell makes the protein in. So the cells, when it wants to make a protein, wants to make a very specific sequence of amino acids. And so it has to really tightly control that production. And we know from our example of sickle cell anemia that if you have I think I always forget this. Hemoglobin was like thousands or more amino acids long, but if you get just one of those wrong, you get a totally different shaped red blood cell. So between the normal hemoglobin and the sickle cell hemoglobin, only one of these amino acids was changed, but you have this drastically different change in this macro scale red blood cell. If you get just one of those wrong, you have a totally different shape. So the point of that illustration is just to show that the primary sequence has to be followed perfectly to make the right protein that does the right job. So that's the primary structure, the primary sequence. The secondary structure is the hydrogen bonding that happens on like a small length scale in these proteins. So you get hydrogen bonding that makes these helices, like we see here, or these sheets that you also see frequently, and those are just due to hydrogen bonding between neighboring amino acids. The tertiary structure is the entire three-dimensional shape of one string of amino acids. So one string of amino acids is going to coil and fold and fold in on itself in a really specific way, and that tertiary structure is dictated by the primary structure. Then you can have multiple proteins that come together to form a quaternary structure. That's what hemoglobin does. So hemoglobin is made up of 4 different subunits that are all jammed together. And so the only way to control this total shape of a big protein like hemoglobin is to carefully control the primary structure, which is one of the things that cells do that we're going to talk about after the exam, is cells can really carefully control what proteins they make. But kind of the point of this was just to show that, you know, we have some behavior of atoms. Atoms want to interact with each other to fulfill their octet role and be more stable. And we can control how we're combining atoms to make different biomolecules. And these different biomolecules, just based on their shape, more or less, are going to have very different roles. So phospholipids and proteins perform very different functions because they have very different structures, and the cell can control what the structures are, which is really one of the cool things about cells, is that they can make exactly what they need to survive. So one more slide in chapter 3, and then we'll go over what potential questions would be. So yeah, maybe I should have showed this earlier on when we first started talking about ATP, but I realized from some of the questions I was getting that it wasn't totally clear what this molecule was. So this is a type of nucleic acid that has 3 phosphates on it. And so when you have these 3 phosphates on here, there's a lot of energy in this molecule, and it stores a lot of energy. And now when you cleave off, or when you remove one of these phosphates, you're releasing some of that energy. So you have this kind of big molecule with these 3 phosphate groups, and just by releasing one of these phosphates off of the ATP, you can release a lot of energy into the environment. And so what we did in cellular respiration is we talked about how you can convert energy from a big carbohydrate like glucose into lots of these ATP molecules to move that energy around in the cell. But it's kind of like the act of putting on the cellular phosphate is like compressing a spring, and like you're adding energy to the molecule. And so this is a type of nucleic acid as well, which I don't think I had ever said to you, which maybe would help you put it in context a little bit. So that was chapter 3. We're like a little over the quarter of the way done the first half of the semester. Is this helpful, or would you rather do something informal and more questions? Or
I think this is helpful.
okay, great. Do you want to talk about like questions that we could have for each chapter?
Yes.
Okay. So it'd be good to know what all the types of biomolecules are. So lipids, proteins, carbohydrates, and nucleic acids. It would be good to know generally what the role of each biomolecule is. So proteins, you could just say catalyzing reactions. Nucleic acids, storing information or storing energy. Carbohydrates, you could say storing energy or structural support. Lipids, is lipids the one I didn't say it? Lipids would be like insulation or energy storage or cellular structure. What else would I potentially ask? Oh, the 4 levels of protein structure. That's definitely important. Does anyone want to go through those one more time? You can. What's the first level? Primary. And what's raise your hand if you want to define primary structure. Yeah. Yeah, go for it.
It's the sequence of amino acids.
Great. What's the second one? Does anyone want to describe it too? I'm trying to conserve my voice.
It's like the local structure, like how the peptide bonds are.
Yeah, well, the hydrogen bonds would be, but yeah, it's like a local structure. Great. What's the third one? Tertiary. Yeah, it's kind of spelled funky. And how do you define that? Yeah, it's like the whole 3D structure of a whole string of amino acids. So you could have like thousands of amino acids all bonded together, and when you kind of let them go and they form their own specific shape, that would be like the tertiary structure of that protein. What's the fourth one? Quaternary. Yeah, I always misspell that one. I have to go back and fix it. And that's how like multiple proteins fit together. Yeah, so that they're not really bonded together. They aren't all in the same string, but they fit together and form like a big protein.
So we want to put the four structures for protein, we ask us what does it do?
I would ask you to list them and just in one sentence define them, just like the level that we talked about would be good.
We were to put those words, what is protein for, like, what is the definition?
Oh, it's a biomolecule that like catalyzes reactions would be good. Yeah. Great. Ready to move on to chapters 4 and beyond? I'm using a lot more color, aren't I?
Yes.
There are like 7 colors in this slide if you count white. White, black, purple. That's what?
So those are all things that we would pay attention to as a chapter 3.
Yeah. I feel like white and black are I'm gonna get credit for those colors, I think. Okay, chapter 4. So we talked about atoms in chapter 2 and how they bond together. We talked about how different molecules can be formed to have very different structures in chapter 3. Chapter 4 is how cells arrange themselves and how they make all those different biomolecules and how they generally are functioning. So we talked a little bit about what a cell is. So cells are the fundamental unit of life, and they're governed by like the 3 parts of cell theory. So the first part is that all organisms are composed of cells. So it's not like most organisms have cells and some are just some other type of life. Like all organisms that we observe have cells in them. And all these cells, in the second part of cell theory, are the basic units of this organism and its function. So not only are all organisms made up of cells, but all of their behavior can really be traced back to cellular behavior. So if we can understand how cells work, we can understand a little bit more about how whole organisms work and how they interact with each other. And the third part is that cells always come from pre-existing cells. So cells are not only making certain biomolecules so that they can grow, but they're also reproducing. And so cells just make other cells, basically. So cells will split into 2 cells, and then those cells will split into 2 more cells. And so we'll talk more about that after the exam. But I think it's important to get a good context of what cells are doing. So there's 2 main types of cells that we talked about. There's prokaryotic cells. So these are simpler and kind of smaller cells, and they don't have membrane-bound organelles. Whereas eukaryotic cells, eukaryotic is like you and me, so human cells, they do have membrane-bound organelles. They're more complex and they're a little bit bigger. Does anyone want to remind me what the importance of membrane-bound organelles is?
Functions within the organs.
Great. So why would a membrane-bound organelle allow you to have a specific function? That was a good answer.
They can maintain a specific type of environment that's conducive to whatever they want to do.
Great. Exactly. Peyton, did you have an answer too?
Same thing.
Okay, cool. So the way biomolecules are formed is just based on the environment that they're in. So if you have a bunch of reactants and you have some enzymes, that reaction is going to proceed and you're going to make some biomolecules. And so by having organelles that have very specific environments, very specific chemical environments, you can control what biomolecules are made or broken down and what they're used to do. So with the eukaryotic cells, which are these bigger cells with lots of membrane-bound organelles inside of them, they have very specific environments in all these different organelles, and so they can do a lot of very specific complex tasks, and they can all come together to do something that's much more complex than you could do in just one big prokaryotic cell. So just wanted to run through a few of the organelles that we talked about. So the nucleus, that's the biggest organelle in the center of the cell. So that's where the DNA is held. So if we look at this top image, we can see a little bit about what DNA is used to do. So DNA is contained in the nucleus, and it sends information out in the form of RNA to the ribosome, and the ribosome is where we're going to make our proteins. But the nucleus, what you have to know for now, the nucleus is the organelle in the cell that holds the genetic information, holds all the DNA.
I know on our quiz you allowed us just to put the name that goes on with it. Will you accept those answers, or are you looking for?
Yeah, those answers would be fine as well. Yeah, so it's like the command center of the cell because it holds like all of the instructions for all of its activities. Then the ribosome, that's where protein synthesis. Sorry, I always forget how bad I am at saying this word. That's where protein synthesis occurs, and that's like the factory of the cell. That's where we're going to make proteins from the genetic information that we get from the nucleus. The Golgi apparatus, that's responsible for like packaging and processing and moving around all these biomolecules. It's sort of like the post office of the cell. I removed the workshop one, the endomembrane system, because I just thought that was too distracting. I couldn't think of a good analogy. I felt bad about workshop. We have the lysosome. Lysosomes are like the recycling plant of the cell. These are like a very acidic organelle in the cell that breaks down biomolecules. So it's going to have a very specific environment inside this organelle that allows it to process and break down biomolecules that would otherwise be functional. And so now it can use all of the resources that were in those biomolecules to make other biomolecules. So it's like the recycling plant of the cell. We have the mitochondria, which we've talked about a lot already, and we'll touch on a little bit later. But again, this is where cellular respiration is occurring. That's where we're breaking down carbohydrates to make ATP. This is found in plants and animals, even though I couldn't get it right before spring break. This is like the powerhouse of the cell. It's always my favorite analogy. We have chloroplasts. These are only in plant cells. These are going to absorb solar energy and use that energy to make carbohydrates. So they're going to take the solar energy and convert it into chemical potential energy. So these are like the solar panel of the cell. Do these sound familiar to everyone? Hopefully nothing's brand new. So then chapter 5 was about another part of the cell membrane. So this isn't really an organelle, it's really just the membrane that surrounds other organelles and surrounds the cell as a whole. So these membranes are made out of what type of biomolecule is it again? Yeah, phospholipids, which are a type of lipid. Great. And so how do phospholipids arrange themselves to make these membranes? What's that?
Polar head, non-polar tail.
Yeah, exactly. So phospholipids have a polar head and they have these non-polar tails. And so when they are put into water, they arrange themselves to have their polar head sticking out towards the water and their non-polar tails sticking into the inside of this bilayer. And when they arrange themselves this way, they can keep the non-polar regions away from the water. And again, water is very polar. We know that because it's composed of hydrogen and oxygen. Hydrogen and oxygen have a very large difference in electronegativity, so water is a very polar substance. So it's going to want to interact more with these polar heads and not so much with these non-polar tails, just like for the same reason that oil and water don't mix. And so you get this bilayer of phospholipids that forms. And this bilayer is going to allow cells to carefully control the environment inside of the cell. Does anyone remember the fluid mosaic model?
They move around.
Yeah, they move around, but which way do they not move around?
They don't move end over end.
I feel like you still read it. What does that mean? They don't move end over end. It's a pretty vague description. Yeah. Right.
They don't flip over on each other.
I'm going to use this very technical pointing instrument. So they'll move this way, or they'll rotate around, but they'll never like flip over that way, or one head will go over the other side. Because in the middle is this non-polar area, and it doesn't want a polar area going through it like that. So the fluid mosaic model is that they're going to move around, so there's a little bit of fluidity to it, so things can sometimes go through because it's not like a rock solid partition between the inside and outside of the cell. It is kind of fluid, but it's not totally fluid. There are some constraints. So some things can get through these bilayers. So these arrows don't really work so well when I cut off the heads. So hydrophobic things, so nonpolar things can go through. Small things, even if they're polar, can go through. But big things and very polar things can't go through. So small things and nonpolar things will go through faster than big things and polar things. And so some molecules, some particles can diffuse through plasma membranes. So the cells do really like communicate with their outside environment like that, like things get in and out of the cell, but not everything. They do have some control. So what would we call this type of membrane if it lets some things through but not everything? Yeah, selective semi-permeable. Guys should be teaching me. So if we know that some things can get through, but not everything. So if we have a difference in concentration between the inside and the outside of the cell, we can get a phenomenon called osmosis. Does anyone remember what osmosis is? June.
The diffusion of water through a semi-permeable membrane?
Exactly. Yeah, that's what osmosis is. It's when water diffuses through a semi-permeable membrane. So if we have a hypertonic solution, or like a solution outside of the cell that's highly concentrated, and this particle, for some reason, either it's very big or very polar, can't get through this semi-permeable membrane, water will then flow out of the cell. When water flows out of the cell, water is really flowing down its concentration gradient, because water is very concentrated here, and water has a low concentration here. So water will diffuse out of the cell. Hypotonic solution outside of the cell, we have the opposite effect. In this effect, in this environment, you'd have water flowing into the cell, and the cell would be swelling. So that's how we can keep track of where water is going during osmosis, based on the concentration inside and outside of the cell. So if you were asked on the exam to define osmosis, you guys would all get 100. If you don't understand it, are there any questions that you would have? Sorry, I keep forgetting to ask when I'm still on chapter 5. Does anyone have any questions about chapter 5 now before we go on to chapter 6? Yeah.
Will index have those things in it?
No, that won't be on there. Well, so no, it won't be on there. But would you like to talk about it anyway? All right, I'll have to start lying then, because we have to talk about all the topics. Yeah. She asked if passive or active transport would be on there, like defining them. All right, chapter 6. Okay, so previous chapters we talked about how atoms are interacting with each other, how they make biomolecules, what cells are made up of, and we talked about some of the organelles, and we talked about the membranes that surround cells, and they surround individual organelles. And so all of that behavior also depends on how energy is moved throughout the cell. And the movement of energy is the study of thermodynamics. So we talked previously about really just how like mass is moved around the cell, like how different molecules move around the cell, and how cells mediate all those changes. But what also matters is the energy within the cell. And so to talk about how energy is moved throughout the cell, we can use these two laws of thermodynamics. So the first law says we're always conserving energy. So we're not going to be creating or destroying energy. We're just going to be changing its form. So cells can't just like be creating energy through some energy source. They just have to change the form of energy that they can get from their environment to do other useful things, like to make other biomolecules or to keep their organization intact, or to grow and reproduce. They need to be taking energy from their environment. But they can't just like be creating energy. They just have to change its form. And so this study of how cells are changing the form of energy is like why we talked about chapters 7 and 8 with photosynthesis and cellular respiration, as we talked about how cells can move energy around and how they can change its form. So the second law of thermodynamics, entropy is always increasing. At every reaction, everything a cell does, it's always going to be losing some energy to the environment. So it's losing usable energy. It doesn't destroy any energy as entropy increases, but it just like loses energy to heat, basically. So every time the cell does something, there's like a little tax that takes some energy that it can't get back. So that means that cells have to really carefully control how they use their energy, because they're always going to be losing some. They're always going to need energy to keep everything functional and intact. So cells have to be getting energy from their environment, and they have to be really careful with how they use it. And so that's going to govern why cells use cellular respiration the way they do and why they use photosynthesis the way they do. So when we talk about energy for reactions and the way whole cells behave, we use this term free energy, or delta G. So the concept behind this is that if there's more products or more energy in the products than there's energy in the reactants, that means we've added energy into this molecule. So if you have, draw something. So if you have some reaction that's like A plus B makes C plus D, these are just some molecules that you have. If there's more energy in the products than in the reactants, that means you have to also have added energy. And so we would say this reaction is like consuming energy, because you have to add energy into this reaction to make it happen. So this would be endergonic. And so this reaction isn't just going to happen spontaneously, it's only going to happen when you add energy into it. In contrast, you can also have reactions that release energy. So as A and B go to C plus D, they can be releasing energy. And so these reactions are called exergonic, and they're like releasing energy to the environment, and these will happen spontaneously. And so what cells will do is they will pair or couple reactions. So they'll put together an endergonic and an exergonic reaction. So that way, when the exergonic reaction happens, it's going to release some energy. That energy can then be used to drive the endergonic reaction. And so that was the idea of coupled reactions. And the reason that's important is because cells are going to use ATP to do this very frequently, because ATP is going to release energy when it breaks down, when it releases that phosphate. And that release of energy is going to be exergonic, and that can be coupled with endergonic reactions that are necessary for the cell to survive and reproduce. But they would be consuming energy.
So that worksheet you gave us, I think this was more for discovery, but it's still that same idea.
Yeah.
Of doing math and figure. Are you going to have stuff like that?
That would not be on there.
Okay, just.
Yeah, I feel like my intentions with that worksheet were good, but it did not work out the way I thought it would. So I'm sorry that we all had to go through that. But we can all learn about coupled reactions, even despite the logistic difficulties of that exercise. So what's important to remember is that you're pairing reactions that are releasing energy with reactions that are consuming energy. But you have to kind of do the accounting right to make sure that everything's going to work out. So like, if you were to release all of the energy in the glucose, which has a lot of energy, to make just one ATP, you'd be releasing a lot of energy, like too much for that reaction to just make one ATP, and so you'd be losing a lot of energy by just releasing to the environment. So you want to kind of do the accounting right to make sure you're using the right energy currency to do the right reactions. So speaking of reactions, so if we look at how energy changes over the progress of a reaction, we see this hump in the middle, and this is related to the activation energy of the reaction. So as reactions go from reactants to products, they pass through this intermediate state called the transition state. So this is an intermediate state between the reactants and the products that has a lot of energy. So that's why this measure of free energy here on the y-axis goes up as we go into the transition state. So the amount of energy associated with the transition state is called the activation energy. So for a reaction to occur, you have to get over this activation energy to get over to the products from the reactants. So what enzymes do, that's really special about them as a biomolecule, is enzymes lower this activation energy. So here you have energy of activation with no enzyme in blue and with the enzyme in the orange. So if the enzyme is there, you're going to lower the activation energy. You're going to make it easier for that reaction to occur. So the easier it is for that reaction to occur, the faster it's going to occur. And so one of the limiting factors with cells is how to make reactions occur really quickly, because sometimes they need a huge number of biomolecules to get something done. And so enzymes are how cells can make this happen. They can make reactions happen very quickly. And in reality, if the activation energy is very high, the reaction won't really happen at all. So enzymes are necessary to make reactions happen quickly in some cases, or just to happen at all in other cases. So enzymes are really crucial for cells' survival and for their healthy behavior. Does this chart make sense to everyone? This little plot? Is that helpful? Does anyone see the connection between, or does everyone see the connection between activation energy and transition state? So they're lined up so nicely like this. Is anyone as excited as I am about this chart? I don't think so.
Do you have a question related to that graph?
No, not. Well, I mean, it's hard to ask. In the description of enzymes, if I asked you to include the terms activation energy and transition state, you would say that enzymes stabilize the transition state to lower the activation energy, which I think I touch on the next slide. Yeah, I do. So we'll talk about that a little bit. So enzymes bind to our substrate. So the substrate is going to go right into the active site of the enzyme, and the enzyme is going to have a very specific shape. And again, enzymes are proteins, so their shape is controlled by the sequence of amino acids in that protein that the cell decided upon, that was like written in the DNA. So that's how they make the right protein to do the right, to catalyze the right reaction. So they have this perfect shape that binds to the substrates, and then they go through this change in shape called induced fit, where they like kind of squeeze around the substrate and like enclose around it to really stabilize both of the substrates. And as they hold the substrates in the right orientation, they're lowering the energy associated with the transition state. So the transition state, again, that's the state where bonds are in the process of being broken and reformed, and so the energy can increase when those atoms aren't fulfilling their octet rule anymore. Because as you break and reform those bonds, in between you're going to have some moments in time where the atoms are not fulfilling their octet rule. And so if they can be stabilized by the enzyme in just the right shape, it can lower the high energy associated with that process. It can kind of help them sort of fulfill their octet rule and make this activation energy a little bit lower. And so after the reaction occurs, the enzyme just lets go of the products and it doesn't get consumed in the process. So that's an important part to remember about enzymes, is they don't get consumed when they catalyze reactions. Did that answer your question about how enzymes work? Again, the key points are transition state. It's between the reactants or the substrates and the products. Activation energy, that's the energy associated with the transition state. And enzymes lower this activation energy. And they don't get consumed in the process. All right, I think that was all for chapter 6. Any questions before we move on?
So we said that entity is moving from, it's essentially losing energy or in energy crisis. Would it also be okay to say that it's like moving towards randomness? I feel like I read that.
Yeah. So that's the link between losing energy to heat and randomness is very connected, but it's hard to. We won't really cover that in this class, but you could put either disorder, randomness, or just like heat, or just like loss of energy. Those will all be fine. I've taken like multiple thermodynamics classes, and I still can't articulate why entropy is like related to disorder and loss of energy, like how that really works. So I'm sorry. People have tried to teach me many times. I just don't.
I don't understand.
Disorderly, chaotic, like none of the molecules will eventually need to split because they're already split, or they
Right.
are already connected, so there's no potential energy left, so that energy just kind of wears off.
Yeah, it's like the measure of disorder is kind of funny. We can talk about it after class. Sorry, I don't want to get sidetracked. But I could send you some interesting articles if you're interested. Okay, so this is taking a little longer than I thought, but I still think it's a good use of time. Do you want to take a break and then do chapter 7 and 8, or just get through the rest of it and then we can just do informal review? Okay, there's just 2 more chapters. It should probably only take like 10 more minutes ish, 10, 15 more minutes, and then we can just do informal review. Is this helpful to everyone? Okay. Oh, sorry, I forgot. I had one more slide. Enzyme reaction rate is controlled by a number of factors. We're just going to talk about 3 right now. Substrate concentration affects enzyme reaction rate. So enzyme reaction rate is going to increase the substrate concentration up until the point of saturation. So up until this point, as you add more substrate in, the reaction is going to keep occurring faster and faster. But after this point, the enzyme is just constantly filled with substrate. So adding more is not going to make it go any faster. It's like if you had like a long line of cashiers, like more people can check out until all the cashiers are always filled, and then just like adding more customers will not make it go any faster. Is that one's better than my last analogy? So then after this point of saturation, increasing the substrate concentration will not increase the reaction rate anymore. So if you wanted to increase the reaction rate beyond that, you'd have to just add more enzymes. So enzyme function also depends on temperature and pH. The pH range of each enzyme is specific to that pH and its function. So we have some enzymes like pepsin, which is found in the stomach and is used to operating in a very acidic environment. We also have trypsin. So these are both enzymes that break down our food, but they're just in different parts of like our digestive tract. Trypsin plays a very similar role to pepsin, but it's just in the intestines rather than the stomach, so it's in a more basic environment. But either of these enzymes put in another environment with a drastically different pH won't function as well, because they'll get, what's the word for what? Denatured. Yeah, my favorite concept. So enzymes have to be in a very narrow range of temperature and pH in order for them to function properly. Does anyone want to explain the temperature dependence of enzymes? Or sorry, why don't enzymes work as good in really cold temperatures?
Because they're not colliding with the substrate.
Great, yeah. So temperature is like a measure of how much molecules are moving around, like how much they're bumping around with other things. And so at very low temperatures, you have a very low number of collisions because your substrate is not moving around as much. And so if your substrate is not hitting your enzyme very frequently, you're not going to have that reaction occurring very frequently. At very high temperatures, though, what happens to the enzyme? Yeah, it becomes denatured because there's just too much energy in all of those little amino acids that are kind of vibrating around, and now they can't be held together with the same bonds anymore. And so it just gets unfolded and has a different shape. When it has a different shape, it has a different function, and it's not going to work as well at what we needed it to do. Okay, on to chapter 7. So hopefully this is one of the most familiar ones in your mind. So just to break down the term photosynthesis. So photo is like light absorption. Synthesis is like making carbohydrates. So photosynthesis, big picture, is using the absorption of light to make photosynthesis. And so we plants absorb that light with a pigment called chlorophyll. And they use the carbon dioxide from the air to make the carbohydrate. So that's the kind of the carbon backbone that makes the carbohydrate. And they get water from the soil to aid in that whole process. So I think one of the interesting things about bio is that all of these things, like the cells have to perform all of these tasks, but they have like very little control over where they get their materials or energy from. So like plants don't have a great energy source besides the sunlight. So they had to grow in a structure that would allow them to use solar energy to do necessary things for those cells to survive. So they kind of have to like respond to their environment in order for them to thrive and like reproduce and become more frequent. So just kind of a schematic of how this process works. So you have carbon dioxide and water going into the plant. You have solar energy, like light energy coming in from the sun into the plant. And we're going to be making glucose and oxygen gas. Again, this is happening only in plants. That's happening in chloroplast. That's the organelle in the plant cells where photosynthesis is occurring, where all of these processes to get all these other resources happen to make the carbohydrates that plants and animals need to survive. So we have two main sets of reactions in this process. So we have the light reactions. This is where we're going to be absorbing the solar energy, taking in the water from the root system, and we make ATP and NADPH. When we send that ATP and NADPH over the Calvin cycle, in the Calvin cycle, we take in carbon in the form of CO2, and we use the energy that we got from the light reactions, that the light reactions got from the sun, and then that's how we make the carbohydrates. So that was the overview of each of the two sets of reactions. So if someone in an exam later this week were to ask you how these two sets of reactions are related to each other, what would a good answer be?
I would say that, the light reaction makes happen and takes in the light from the sun, and the Calvin cycle uses the sun, that the light reaction took in to make carbon dioxide.
To make carbohydrates from carbon dioxide. Yeah, but it takes the energy in the form of NADPH and ATP from the light reactions to make that happen. Yeah. So that's how we could say they're connected, is the light reactions send energy over to the Calvin cycle in the form of NADPH and ATP. And then the Calvin cycle uses that energy, and it uses carbon in the form of carbon dioxide to make a carbohydrate.
Will you have like a diagram like that, or is it just going to be short answer?
It would just be short answer. I'm going to ask you to tell me how these are connected, and that would be a good answer. That energy goes from one cycle to the other.
Could you repeat what you just stated?
Definitely. No, I'm happy to reiterate it. The light reactions send energy in the form of NADH and ATP over to the Calvin cycle, so the Calvin cycle can use that energy to make carbohydrates from CO2.
So basically, we're making
Yeah, that would be fine. So now we're going to talk a little bit more in more detail about how each reaction works. So the light reactions, we have energy coming in, we have solar energy coming in, and that's going to make like a high energy electron. So we're going to have energy going to these pigments that are in the cell. Mainly chlorophyll is the pigment that's absorbing the solar energy. It's going to absorb that solar energy, and it's going to excite an electron. So now you're going to have like a really high energy electron. That high energy electron is going to like flow down this electron transport chain. So as it goes from each part of the electron transport chain to the next, it's going to lose a little bit of energy, like a ball rolling down a hill. It's just going to kind of flow down this chain. And that electron is going to end at the NADPH. And so now you're going to have an energy molecule that has some of the energy from the high energy electron now on this NADPH, and now you can send that molecule over to the Calvin cycle to do some useful work, to like carry some energy. Also, as this electron is going down the electron transport chain, you're pumping protons up their concentration gradient. So you're going to have a really high concentration of protons in this intermembrane space in the organelle here. And so when you have this high proton gradient, they're going to want to flow out. They're going to want to flow down their concentration gradient and like release energy that way. And as they flow down their concentration gradient, they're going to flow through this enzyme called ATP synthase. So ATP synthase is an enzyme that uses energy from this flow of protons down their concentration gradient to synthesize ATP. So we saw like a short YouTube video on this. As the protons flow through here, they basically spin this around. They spin this enzyme around, and as it spins around, it's going to be making ATP. So it's going to be combining ADP and a phosphate to make an ATP as protons flow down here. And the protons got here in such high concentration, again, from this electron transport chain. So this is similar to what we'll see in chapter 8 as well. Any questions about the electron transport chain?
So the electrons are essentially doing two things. It's turning ADP, and at the same time it's moving the protons, which create ADP.
Yeah, exactly. Yeah. So the electron performs two tasks, like you said. It ends with some of its energy on this NADPH. This is now a high energy molecule that carries energy around throughout photosynthesis. And they also pump protons up their concentration gradient, and that's then used to make ATP.
So like, how do we differentiate what's happening? Like, is it the same concept with the light reaction and then cellular respiration and the electrolysis?
It's very similar. In cellular respiration, rather than the electron ending at NADPH, it ends at an oxygen. And so it just releases more energy because oxygen really wants that electron.
So the only difference is that what it is.
Correct. Yeah, but it still uses like the same driving force and the same enzyme to make ATP. I think some of the proteins in the photosynthesis, like in the chain, might be different, but that's not really important for this. All right, so just a brief overview of the Calvin cycle. So we have 3 steps. So we talked a little bit about these steps before. So the first step is the fixation of carbon dioxide. So you have carbon dioxide that's out in the atmosphere, and you want it to bond to a molecule in the plant, which is not very common. Carbon dioxide doesn't just readily bond to a lot of molecules. So you have a really specially designed molecule that's going to attract this carbon dioxide. And that's what we're going to call this RUBP. Apparently, I was reading, some people call it RuBP, which is like a lot more fun to say. So I think I might just call it RuBP, but that does not like distract anyone. No one cares. No one's really like particular feeling either word. So this RuBP, I'm going to call it RUBP. That's too much change. So this RUBP is going to bond to this carbon dioxide from the atmosphere, and you're going to go from a 5-carbon molecule to making this 6-carbon molecule. So that's the fixation of carbon dioxide. So you had a 5-carbon molecule, and it's going to just bond to some carbon dioxide that was out in the atmosphere, which is like not an easy thing to do. It's not very common. Now you're going to make this 6-carbon molecule. Then your second step is the reduction of your carbon dioxide. So now you have a 6-carbon molecule, and you're going to break it up into 3-carbon molecules in the form of G3P. So G3P is like a really high energy, valuable molecule for the plants. So they're going to send some of that G3P out of this process to be used to make carbohydrates or other things in the plant. And they're going to send the rest into regenerating RUBP, which is the final step. So the final step is you regenerate what you started from. And so this way you take in carbon from the atmosphere and you let the same amount of carbon out to be used to make carbohydrates. But everything within this cycle is always being remade, like replenished. So it's more sustainable. So that was our summary of the Calvin cycle.
Do we need to know a specific amount of G3P that's going to be reused to create RUBP?
No, you don't have to know. No. Oh, sorry. Yeah. No, I would ask you what the 3 steps of the Calvin cycle were.
Okay.
Yeah.
You also still like say what happened?
Just briefly described. Yeah. Just that level of detail would be fine. Nothing more than that is required. So I wanted to also talk a little bit about an alternate form of photosynthesis. So most plants don't undergo this form of photosynthesis, but in really hostile environments that are very dry or very hot, you have another form of photosynthesis called CAM photosynthesis. This is where plants are going to let in CO2 only at night, and they're going to close their, so in the plant we had this stoma, which was the little hole that let in CO2, so it could bind to the RUBP. So those holes are going to open at night, and they're going to close during the day. And so when they close during the day, they're going to reduce the amount of water loss or evaporation that the cell is going to undergo. And so that's going to make these plants a little more apt to survive in really dry or really hot environments. But they still have to take in their CO2, so they just open these stoma at night. And so this gives some plants an advantage in really hot, dry environments. So the important part about that, I think, is to realize that organisms and cells have really clever adaptations to survive, and they can make a lot of changes to be able to thrive in different environments.
Do they detect day and night by the amount of solar energy they absorb?
That is a good question. I don't know, or maybe they like have some sort of circadian rhythm of some sort.
Wouldn't that be confusing though? Like with the seasons and...
Yeah, that's a great. I don't know how they know when to open and close. It could be solar energy. It could be like some sort of heat thing or like water loss thing. I don't know. So good. The cool thing about bio is it gets like endlessly complex. You can just like dig into any of these topics, and it's really easy to get to a point where not many people know all the answers. It's just like a lot of mysteries involved. Not that like no one, I'm sure a lot of people know how this works, but like it's just there's a lot of detail out there to really understand how things happen. So last chapter, so we're going to talk about cellular respiration. So photosynthesis, again, only happens in plants. Cellular respiration happens in plants and animals. So the goal of cellular respiration is to break down carbohydrates, mainly glucose, to make ATP. So here we're going to break down these carbohydrates and convert the energy from being in glucose, which has a lot of chemical potential energy, to being in ATP, which are a much smaller unit of like energy currency. So again, you can make like 36 to 38 ATP from one glucose molecule. So which molecule do you think has more energy in it, glucose or ATP? Glucose, great. Because as it breaks down, you make like nearly 40 ATPs. And you're not creating any energy in the process. And you know as this whole process happens, you're losing some to entropy. So there's a lot of energy in glucose. Some gets lost in the environment, and it gets broken up into lots of little ATPs. So the 4 steps of cellular respiration, we have glycolysis, preparatory reaction, citric acid cycle, and the electron transport chain. So we're just going to briefly go through what happens in each of those steps. So if we just look at the energy first, I hope this table makes more sense. I tried to make some changes. I also added in the numbers, like before each molecule. You don't have to know those, but I thought maybe it would help you keep track of where everything's going. So the initial form of energy in cellular respiration is just in this glucose molecule. So glucose has a lot of energy in the bonds of this bottom molecule. And so you're going to break that down, and you're going to get some ATP and some NADH in glycolysis. And then in this next step, you're going to get some more NADH and no ATP. Citric acid cycle, you're going to get some ATP, a lot more NADH, and some FADH2. And then your final step, you're going to use all of that NADH and FADH2 that you had made before to make ATP. And so if you look at it kind of in total, you see that we're going from glucose to making ATP because you have the same amount of NADH here in your initial and your final.
Especially like the acid cycle, if you wanted to know like how many exactly, or if we just said the final result is ATP and ADH and 2 FDH2.
Yeah, you don't have to use any of the numbers. Yeah.
Will we see something like that on this table?
No.
Or more explanation?
I would ask you to name what the 4 steps are and how that, what's happening. So that's just the energy, but also we have what's happening to like the carbon. So we kind of look at them separately to see how they're both interact with each other. So the initial source of carbon is glucose. So same with the energy balance. And then glucose gets broken down into these 3 carbon molecules called pyruvate. So it just gets broken in half. So in the process of breaking carbon in half to make these 3 carbon molecules, you're creating ATP and NADH. In the preparatory reaction, you're going to take these 3 carbon molecules, you're going to break 1 carbon off, that's going to go to carbon dioxide, and you're going to have a 2 carbon molecule. That's acetyl-CoA. So preparatory reaction, we go from pyruvate to acetyl-CoA and carbon dioxide. Citric acid cycle, you're going to go from these 2 carbon molecules, you're going to break them in half, you're going to get carbon dioxide. And now you've taken all of the carbon that was originally in glucose, all 6 of these carbons, and you made 6 carbon dioxides. Sorry, I should say 6 carbon dioxide. That'll make that more clear. So overall, what's happening is we have glucose and we get ATP. We have glucose and we get CO2. So we're taking all of the carbons and glucose, breaking them down into CO2. We're taking as much of the energy as we can from the glucose and we're making it into ATP. Does that overview, does that make a little more sense or less sense? Or should I have showed this the first time we went over it? Or that would have just been more confusing? So all that's really important to know is that the electron transport chain makes the most ATP, and it does it by using NADH and FADH2. And all of the ATP, all the energy comes from glucose, and all the carbons from glucose go to carbon dioxide. But the first three steps don't really make any ATP. They mainly make NADH and FADH2. That is then used in the electron transport chain to make ATP. Any questions? All right, so let's just run through this one more time, just to clear up any confusion. So this mnemonic is going to be helpful as we go through this. Oxidation is our loss of electrons, oxidation, loss of electrons. Reduction is when we're gaining electrons. So NADH and FADH2, they arrive at the electron transport chain with a high energy electron. And so they're going to lose that electron. Losing that electron is oxidation. At the end of the electron transport chain is the oxygen, which is going to receive the electron. And that gaining the electron is the reduction. Sound good?
Do you need to know which one's the reducing agent?
No, I mean, it's just important to know that they happen in pairs. I won't use these terms, reducing agent and oxidizing agent, but when one thing's being reduced, something else is being oxidized, so the reactions are coming in pairs. I think this is our last slide. So if oxygen isn't present, so oxygen is the final acceptor at the end of the electron transport chain. If oxygen isn't present, we can't go through all of cellular respiration, but we can go through the first step, which doesn't require any oxygen. So that would be glycolysis. So in glycolysis, we're going to be making some ATP from, as we break down glucose. But with glycolysis alone, we're converting all this NAD plus into NADH. And so what fermentation allows us to do is that it allows us to recycle this NADH back to NAD plus, so that we can have a cycle that can just keep going and is more sustainable. So now we can make some ATP from our glucose without totally depleting our stores of NAD plus. The downside to fermentation is that we produce very little ATP for a whole glucose molecule. So we only produce 2 ATP rather than like nearly 40, which we had for cellular respiration. So it's less efficient, and it also produces a harmful byproduct in the form of either lactate or alcohol. But sometimes if an organism really needs like a burst of energy, either like to run after prey or to like survive being chased by a predator, it will need to produce more energy than it has oxygen to do so. So it'll have to undergo fermentation to make that happen. All right, that was the whole review. I'm sorry that took a little longer than I thought. I'll put it online and the recording. Does anyone have any questions before we go?